Hello, everyone, and thank you so much to the conference organizers for inviting me to speak today. I will be talking about one of the most exciting areas emerging in science, the microbiome, and its role in patients with colorectal cancer. These are my disclosures, and this is a brief outline of my talk. I will first review some basic background about colorectal cancer as well as the microbiome, then talk about selected high-impact research that we and others have been involved in with the microbiome, and then talk about some of the studies that we are currently working on to try to answer some of the outstanding questions that still remain. And then finally end with some conclusions and future directions. As many of you know, colorectal cancer is a huge problem in the United States. In 2021, it is going to be the third leading cause of cancer in both men and women, with over 150,000 new diagnoses expected. When you combine men and women together, it is actually going to be the second leading cause of cancer death, with over 50,000 people unfortunately expected to die of this disease this year. Fortunately, there have been steady improvements in survival, and that's largely because there have been multiple discoveries of new drugs to treat patients with metastatic disease. These include 5-fluorouracil, which was the only drug we had for a really long period of time, followed by newer chemotherapy agents that have improved efficacy, and then some more targeted therapies now that have emerged that have significantly improved survival from this cancer. In fact, in 2021, these are the available FDA-approved, mostly, treatment options for patients with colorectal cancer. You will see here that we are now starting to get into options for targeted therapies, including BRAF-targeted therapies for those harboring BRAF mutations, and HER2-targeted therapies for those who have tumors that overexpress the HER2 protein. And then very exciting, we now have some immunotherapy options for a small proportion of patients with a specific type of metastatic colorectal cancer called microsatellite instability high, or MSI-high. You can see here in this study of patients who were receiving pembrolizumab that for patients with MSI-high or dMMR colorectal cancer, there are significant responses to immunotherapy in these patients. This is a rare subtype of colorectal cancer affecting less than 5% of all patients with metastatic disease. But in those patients, the responses have been significant and promising and durable. In this spider plot here, you can see the light blue lines show a significant improvement in biochemical response as well as radiographic response for patients with metastatic colorectal cancer who are MSI high and treated with pembrolizumab. Unfortunately, for the vast majority of colorectal cancer patients who have proficient MMR or MSS metastatic colorectal cancer, there are no responses to immunotherapy. You can see here that all of the proficient MMR patients had increase in the size of their tumors and no evidence of shrinkage in response to these newer immunotherapy drugs. And this is a huge problem and a huge research priority for researchers in the field. The biggest challenge now is that most colorectal cancers remain cold. And what we mean by cold is they're resistant to intrinsic anti-tumor immunity mechanisms that our bodies have, and they are resistant to the ability of existing immunotherapy drugs to convert these cold tumors to hot tumors who then respond to immunotherapy. And one of the leading hypotheses for why it is that colorectal cancers are cold tumors rather than hot tumors is that the colon is actually one of the most densely populated microbial ecosystems on the planet. There are trillions of organisms called the microbiome that are present in all of our guts today. Here are some basic facts about the microbiome. So again, the microbiome is the community of microorganisms that reside in our bodies, the vast majority of which are in our intestines. There are up to 100 trillion microbial cells in our body that way outnumber the number of human cells in our body by 10 to 1. They also express many more genes than human genes, 100 to 1 in terms of the number of genes expressed by microbial organisms as opposed to human cells. And this accounts for significant variation between individuals, where it is very plausible that perhaps differences in the microbiome may be influencing each person's risk of certain chronic diseases, including cancer. We are now beginning to understand that the makeup and the number of organisms in our microbiome may significantly contribute to multiple processes in our body, including being involved in obesity and promotion of fat storage, promotion of things like angiogenesis, which is the process by which blood vessels are formed. The microbiome has significant implications for the composition of our immune system. It is involved in synthesis of essential nutrients that we all need. It helps in metabolizing drugs that we ingest for a variety of different conditions. It can affect the nervous system. It can break down the food compounds that we all ingest. And of course, it can help in terms of protecting us from infections as well. Now, what are some of the mechanisms by which the microbiome may be involved in cancer formation? There are two types of bacteria that we think are possibly relevant. One are bacteria called driver bacteria, which can potentially directly cause cancer, either through the metabolites that are produced by these bacteria or by toxins that we know can be damaging to DNA and to some of the processes that lead to cancer formation. The other types of bacteria that may be involved in our microbiome in terms of causing cancer are bacteria called passenger bacteria, which are there just along for the ride because this environment is conducive to allowing them to expand. Both types of bacteria can then interact with processes in each person's cell and affect the signaling pathways that are often implicated in why colorectal cancers develop, including pathways like the Wnt beta-catenin signaling pathway and processes that damage DNA that can lead to cancer, that affect the inflammation status in our bodies, and that affect our immune systems. We know that in addition to the microbiome contributing directly, potentially, to the formation of cancer, that the microbiome is also significantly shaped by diet and lifestyle factors and other environmental exposures, which they themselves can also contribute to cancer. The immune system also significantly shapes the microbiome and vice versa, as we just discussed. And we know that diet can also shape our immune systems, and the immune system itself can lead directly to cancer formation as well. So it is likely that the processes leading to colorectal cancer involve a complex interplay between diet and lifestyle, the microbiome, and the immune system, all of which are likely working together to increase the risk of cancer. We are now also starting to understand that the microbiome may also be very important for affecting responses to cancer treatment, especially responses to immunotherapy. In a series of high-profile papers published about immunotherapy effectiveness in other types of cancers, such as melanoma and lung cancer and kidney cancer, multiple organisms in the microbiome have been identified that predict how likely it is that immunotherapy will work in these types of cancers. Currently, there are no large-scale studies about the effectiveness of immunotherapy and what factors in the microbiome may affect that for colorectal cancer. And that is one of our research projects that we have going on that will carry huge importance for understanding whether we can modulate the microbiome to improve responses to immunotherapy in patients with colorectal cancer. So let's go through some of the research studies that have been done that helped improve our understanding of the role of the microbiome in colorectal cancer. Here's a brief timeline of some of the highlights and important papers regarding the microbiome in colorectal cancer. One of the first important papers was published over a decade ago in 2009 that identified a bug called Bacteroides fragilis as being important for the development of colorectal cancer. Subsequently, multiple studies have shown that a bacteria called Fusobacterium nucleatum also seems to be very important for initiating the processes that lead to colorectal cancer, and potentially also affecting survival of patients who do harbor this bacterium in their microbiomes. We have also shown that Fusobacterium can directly lead to colorectal cancer formation, as well as another organism called PKS positive E. coli, which is a paper that I will talk about in a little bit more detail. Let's go into some of the papers that we have highlighted in the previous slide. One important finding that has recently come out in a couple of meta-analyses performed in 2019 include that there is a consistent set of core bacteria that do seem to be enriched in stool samples from patients with colorectal cancer compared to stool samples from healthy controls or people without colorectal cancer. Some of these bugs are familiar, such as Fusobacterium nucleatum, which we just talked about, Parvomonas, Peptostreptococcus, Clostridium, Prevotella, as well as a variety of other organisms that have been shown in other separate studies to be involved in the pathogenesis of this disease. This certainly points to a huge potential of the microbiome to perhaps be a tool for early detection as well as screening. We can potentially combine the identification of these organisms that seem to be associated with colorectal cancer with existing stool tests, for example, that can potentially increase the sensitivity of screening tests to detect colorectal cancer and perhaps prevent colorectal cancer. Efforts are also underway to investigate whether there are specific bacteria that are also associated with adenomas, which are the precursor lesions that lead to colorectal cancer. And if we can identify a core set of bacteria associated with adenomas as well, it may also help with identification of patients who may need colonoscopy screenings to try to remove those adenomas for cancer prevention. However, before this is ready for prime time as a screening test, multiple challenges still remain. And those include that some of these tests are still not high enough in terms of sensitivity and specificity to be deployed as a population-wide screening tool. And so efforts are still needed to improve the accuracy of microbiome detection for screening purposes. Currently, these microbiome identification tools seem to work best for picking up colorectal cancer in patients who have stage 3 or 4 disease. And if we want to do a better job using the microbiome for early detection and screening, we really need to get it to work better for earlier stage disease, namely people who have stage 1 or 2 colorectal cancer. We also need to prove still that the organisms that are associated with colorectal cancer as opposed to healthy people are consistent across different populations around the world, as well as different subgroups of patients according to race and ethnicity, for example. And finally, we know the microbiome can be very influenced by diet, as well as medication and other medical conditions. And how do those affect the accuracy of the ones that are associated with colorectal cancer? Finally, the study, for example, that led to the identification of these core bacteria as being associated with colorectal cancer were done with extremely sophisticated and expensive sequencing tools. And if we are to have a screening test, we need to find a more cost-effective way to identify these bacteria that can be deployed on a population-wide level. So certainly challenges remain, but this is a very promising area in terms of improving our ability to screen and detect colorectal cancer early using the microbiome. This is an important paper that just came out recently from several of our collaborators who we work with in the setting of a large grant award that has shown a direct causal effect of bacteria in terms of leading to DNA mutations that can cause colorectal cancer. In this study, investigators injected this bacteria called PKS positive E. coli, which produces a toxin called colibactin, and they injected this organism into human intestinal organoid models. You can see here the bacteria directly interacting with these organoid models. We show that after injection and co-culture of these organisms with organoids, DNA damage is induced by this bacterium. And that can be seen here by the fluorescent pink areas inside the nucleus of this organoid. In contrast, when E. coli was injected that is not able to produce colobactin, no DNA damage is detected. Similarly, when a benign dye is injected with these organoids, no changes in the DNA are seen. This is a positive control, mitomycin C, which is a chemotherapy that is known to damage DNA. And you can see here that when organoids are injected with this chemotherapy agent, DNA damage occurs as well. So this is direct evidence that the effects of an organism in our microbiome can actually damage DNA in colon cells and lead to a distinct signature of mutations that this group then subsequently identified in metastases of colorectal cancer patients. This opens up possibilities for screening and early detection because it certainly suggests that individuals who harbor this organism in their microbiomes may be at increased risk of developing colorectal cancer down the road. And if there is a way to pick up those people earlier and identify them for screening, perhaps we can make headway in terms of decreasing the incidence of colorectal cancer in these high-risk individuals. It also leads to opportunities to perhaps target and eliminate and treat this organism with targeted antibiotics to hopefully get rid of them and their potential to lead to colorectal cancer in our microbiomes. Another organism that we have done a lot of work on is Fusobacterium nucleatum, which has been found in much higher abundance in patients with colorectal cancer compared to people who do not have colorectal cancer. We have also done studies that the amount of Fusobacterium in the microbiome differs depending on what part of the colon you look at, where Fusobacterium seems to be enriched in right-sided colon tumors as compared to left-sided colon tumors. When we compared patients who had cancers that had Fusobacterium compared to patients with colorectal cancers that did not have Fusobacterium, we saw that those patients who did have Fusobacterium in their tumors had significantly worse survival compared to patients who did not harbor this bacterium. This certainly suggests that this bacteria may be doing something to these tumors that make them more aggressive and perhaps less responsive to treatment. We now also have some data that Fusobacterium may be leading to this worse survival by interfering with anti-tumor immunity, where we studied Fusobacterium high colorectal cancers and found that they had lower levels of infiltrating immune cells in their tumors. And we know that the more infiltrating immune cells are found in colorectal tumors, the better the survival is of those patients. So Fusobacterium may be suppressing immunity against colorectal cancer. In a study that we did comparing tumors who had Fusobacterium in the colon compared to the liver metastases of those same patients, we actually found that you can detect Fusobacterium even in the liver metastases. This suggests that Fusobacterium actually invades colorectal cancer cells and may travel with those cells as they disseminate to distant locations to metastasize. You can see here that we were even able to culture out live Fusobacterium organisms from both colon primary tumors as well as liver metastases in the vast majority of patients whom we studied. When we looked at the overall microbiome found in colon primary tumors from these patients and their matched liver metastases, the community of microorganisms in the microbiome was remarkably similar between the colon primary tumor and the liver metastasis. And this suggests that organisms in the microbiome may actually be invasive or perhaps stuck to cells that are right in the tumor microenvironment that then travel with the cancer cells as they metastasize. This has a suggestion that perhaps the microbiome is implicated in metastasis as well as development of the initial cancer. When we then looked at whether or not the microbiome and Fusobacterium specifically travels with cancers as we implant them successfully into different mice for living tumor models, we also found that Fusobacterium does persist in these successive generations of patient-derived xenograft models of colorectal cancer. again suggesting that the fusobacterium is probably invasive into the colorectal cancer cells, which is why they persist and are found in these successive generations of living tumor models. Again, when you look at the composition of the microbiome in these xenograft models compared to the original tumor from the original patient, you can see that the composition of the microbiome here is extremely similar when you look at the primary tumor compared to the first generation of this xenograft model in a mouse, as well as the third generation of this xenograft model in a mouse. Again, suggesting that the microbiome may have a direct role in the pathogenesis of this disease and in metastasis. We next looked at whether or not if you try to treat this bacterium, fusobacterium, with an antibiotic, whether or not that can actually help tumor growth. And indeed, when we treated a model that did not have fusobacterium in it with metronidazole or flagyl, which is an antibiotic that does have activity against fusobacterium, you can see that there's no impact at all on the rate of growth of the colon tumor in this mouse model. On the other hand, when we looked at a mouse model of a colon tumor that did have fusobacterium in it and treated them with this antibiotic metronidazole, you can actually see a slowing of the growth of the colon cancer just by using this antibiotic to treat fusobacterium, which again implies that fusobacterium may be playing a central role in tumor growth, and that if we can find novel ways to treat this bacteria with more specialized antibiotics, we may actually also significantly impact and prevent the growth of the colon cancer itself. And so all this data really point to and support an important role of the microbiome in colorectal cancer. But many questions remain, including the ones listed here, which are the exact microbes that are most important for colorectal cancer and that indicate that somebody is at high risk of developing colorectal cancer? When does the microbiome emerge as a key player in the process of cancer development? Who are the individuals who are at risk of microbiome-related colorectal cancer? And where are these microorganisms? Are they invasive into the cells as we think they are for fusobacterium, or are they affecting or on immune cells? This is still a question that is not answered. What are the mechanisms by which the microbiome promotes colorectal cancer? Does it affect the cancer cells directly? Does it damage DNA, as we saw with PKS positive E. coli? Or is it affecting the immune microenvironment? Can the microbiome be modulated to improve response to existing treatments, especially immunotherapy? And then can we use knowledge of the microbiome and how it might be acting in colorectal cancer to develop novel treatments, such as targeted antibiotics for treatment of colorectal cancer, or ways to accomplish chemoprevention in people who are at high risk of developing colorectal cancer? And obviously, our interest in trying to answer all these questions is so that we can improve the survival of colorectal cancer patients, as well as decrease the risk of developing this disease for people around the world. So how are we trying to design some research studies to better understand the microbiome in colorectal cancer? As we just heard, the colorectal cancer is likely a very complex, multi-stage and multifactorial condition that is affected by genetics, diet and lifestyle, and other influences in the environment. Obesity and inflammation play a significant role in our risk factors for developing colorectal cancer. And then, as we just heard, the microbiome is also critical in the pathogenesis of this disease. And it's likely that all of these factors are interacting together to lead to the development of this cancer in the colon. So in order to study the role of the microbiome, you not only have to collect tumor and stool samples to look at the microbiome, but you also need data and information from all these other factors that are involved in the disease process. And that is the problem with current cohorts that are out there. Many have small numbers of patients, and you really do need huge numbers to be able to come up with definitive answers to some of these important questions. Many also only have a single time point of collection of samples, including stool, DNA, as well as tumor samples, which is not helpful if you want to understand how the microbiome is changing in response to different treatments. We also often don't have good information on the genomics or genetics of patients with these cancers to correlate with the microbiome, or good enough data on the clinical features of the patients with this disease in the cohorts, or assessment of diet and lifestyle factors in a validated fashion. And finally, we do need diverse patient populations if we are to understand if some of our findings are applicable to patients of all racial and geographic subgroups. And so one very exciting project that we are involved in is a Cancer Grand Challenge grant team called Optimistic, which stands for Opportunity to Investigate the Microbiome's Impact on Science and Treatment in Colorectal Cancer. The overall principal investigators of this grant are Matthew Meyerson and Wendy Garrett, both here at Dana-Farber Cancer Institute with me. And I am fortunate enough to be part of this international dream team of researchers and scientists who are really working to try to answer these questions that remain about the role of the microbiome in colorectal cancer and how we might use the microbiome to improve treatment and prevention. Aim one of this grant is to really map the microbiome in colorectal cancer, both what the role of it is in cancer risk, how it is influenced by epidemiologic factors like diet and lifestyle, and where it's located in the tumor as well as in the tumor microenvironment. Aim 2 is focused around how we can use the knowledge about the microbiome to improve treatment and prevention. And this is trying to develop better laboratory models for studying the role of the microbiome and new ways to target the microbiome, trying to understand the role of the microbiome in treatment response in our colorectal cancer patients, and also how we can manipulate the microbiome in new ways to come up with novel treatments for patients with this cancer. And so the project that I have within this large dream team is to organize and start a large international prospective cohort study called Microcosm, which stands for Microbiome of Colorectal Cancer, Longitudinal Study of Mechanism. Several different international sites are already involved in this project, including us at Dana-Farber, St. Luke's Cancer Institute in Boise, Idaho, UCSF in San Francisco, Vall d'Hebron Institute of Oncology in Barcelona, Spain, as well as the largest cancer center network in Brazil called Oncoclínicas. We are actively looking to accrue more centers to participate in this study, where we will collect tissue, blood, and stool from all colorectal cancer patients, and then look at treatment outcomes as well as diet and lifestyle factors and try to correlate all of this with the microbiome. We plan at patient enrollment to collect all of the different samples and clinical information, including tumors, DNA, serial blood samples, serial stool samples, and diet and lifestyle information. And in order to understand the role of the microbiome in influencing treatment response, we will want to collect these samples pre, on, and post-treatment as patients progress through their various cancer therapies. We will also be doing pre, on, and post-tumor biopsies to better understand what is happening in the tumor itself in response to treatment, as well as once a year reassess diet and lifestyle to see whether changes in diet and lifestyle are also impacting the microbiome and whether that can influence response to treatment. We are interested in 3 different cohorts of patients in this microcosm study. The first are patients who are being treated with immunotherapy. And currently we have 115 patients already enrolled with pre, on, and post stool and blood samples, as well as diet and lifestyle questionnaires completed. The second cohort of patients are young onset colorectal cancer patients, where we do think the microbiome may be playing a major role in why this incidence is increasing in young people. And then the third cohort are patients treated with 5-FU-based chemotherapy, because we do know that the microbiome plays an important role in metabolizing IV drugs as well as oral drugs, and may be important in whether or not patients have side effects from these chemotherapies. Overall, we now have over 500 patients enrolled on this project and have already started to assay some stool samples to try to get preliminary information on how the microbiome may be playing a role in each of these different patient groups. I want to focus also on some of our work on young onset colorectal cancer. As we have heard in patients under the age of 50, the incidence of colorectal cancer is rising and mortality is starting to increase as well. And it is estimated that by the year 2030, colorectal cancer is going to surpass breast cancer as the leading cause of cancer death in people between the ages of 20 and 49, which highlights the importance of better understanding what the underlying causes are. A good first step in helping to reverse these trends, hopefully, is that the United States Preventive Services Task Force did recently lower the guidelines to initiate colorectal cancer screening down to age 45 from age 50. This will certainly help to prevent and detect more cancers earlier in young people, but it certainly doesn't help the very many young patients who we see and care for in their 20s and 30s, and for whom this cancer is rising the most rapidly. So this points to the urgency of better understanding the underlying risk factors and causes so that we know who are the high-risk young individuals to target for earlier screening. As part of our Young Onset Colorectal Cancer Center at Dana-Farber Cancer Institute, which was launched in March 2019, we also launched a separate flagship research project called the Beyond CRC Project, which stands for Better Understanding of Young Onset Colorectal Cancer. We are extremely grateful to the Colorectal Cancer Alliance for funding a research project that we think will really help us to better understand what the differences are in the microbiome between young and older patients. The first aim of this grant, that is generously funded by the Colorectal Cancer Alliance, is to study patients across the spectrum of young age. Patients who are in their 20s or younger, in their 30s, in their 40s, and also older onset patients, and then to collect stool samples from all of these different age groups so that we can analyze the stool and better understand what the differences are in the composition of the microbiome and the diversity of organisms studied, and whether these significant differences between decades of young age may be contributing to why cancers seem to be more aggressive in the very youngest patients. Because our Beyond CRC project also has information on clinical features and treatment, diet and lifestyle, as well as genomic data, we'll also be able to correlate these differences in the microbiome with these other very important factors to get a comprehensive view of what might be causing young onset colorectal cancer and how it might be different from older patients with this disease. The second aim is, again, to use the Beyond CRC project samples from young patients compared to older patients, and then to transfer the microbiomes of young versus old patients into what we call germ-free mice. These are mice who are bred without their own microbiomes. And so when you transfer the microbiomes from human young patients versus human old patients, we can directly assess the effects of the human microbiome on the colorectal tumors in these mouse models. Again, our goal is to compare differences in tumor growth of these colorectal tumor mouse models and whether there are differences in the immunity against these cancers imparted by the microbiomes of young versus older patients, to directly understand what features of the microbiome are contributing to the differences that we think exist between the aggressiveness and treatment response of colorectal cancers that happen in younger people versus older people. And again, the great benefit of having the Beyond CRC project's comprehensive data and biospecimen collection is that we will be able to correlate features of the microbiome with diet and lifestyle, as well as genomics. So I look forward to updating you as the years go on and our project advances on the preliminary findings that we see. And then finally, it is critical that we accelerate the pace of research on colorectal cancer to better understand the role of the microbiome. And that was the huge impetus for us to launch the Count Me In Colorectal Cancer Project. Count Me In is a digital social media platform that directly engages and partners with patients no matter where they are in the US and Canada to help us accelerate the pace of research. This will allow patients to consent and send in their clinical information and specimens all through an online portal. This will allow us to reach patients that are more diverse and have a sample size that is more inclusive in regards to racial and ethnic groups, socioeconomic status, as well as different geographic areas in North America. And importantly, it will allow us to continue this research even during pandemic times when travel to some of these larger centers doing this work may be limited. And also it helps for patients who don't necessarily have the resources to travel to these centers to participate in research, to participate from home. We are excited to announce that the Colorectal Cancer Project on Count Me In did launch on September 14th. This is the website link. And any patient with colorectal cancer, no matter when and at what age of diagnosis, is eligible. So I definitely encourage patients out there to sign up and participate and really partner with us in trying to better understand the impact of the microbiome. So in conclusion, we know that the gut microbiome plays a central role in the development of colorectal cancer. Specific microbes, including Fusobacterium and PKS positive E. coli, have been directly implicated in causing colorectal cancer in laboratory studies. More studies are needed in human patients to understand the role of the microbiome in screening, early detection, and treatment. Many research efforts are ongoing to help try to figure out how we can change the microbiome for cancer prevention and treatment. And patient partnership in these efforts is critical if we are to accelerate the pace of our work. The potential of the microbiome is vast, including understanding what biomarkers in the microbiome could be used for screening, as well as predicting clinical outcomes and responses to treatment. And the types of markers include looking at microbial genes and metabolites, using samples from the stool, the mouth actually, blood, and tumor tissue as well to identify these biomarkers. And then very exciting, how can we change the microbiome to help improve treatment, especially to immunotherapy, as well as for prevention purposes to identify high-risk patients for screening. With that, I want to thank the many different collaborators who have helped in this work, including my colleagues at Dana-Farber Cancer Institute and the Harvard Chan School of Public Health, at the various sites participating with us in the CRUK Grand Challenge project, as well as Beyond CRC. Our many patient advocates who have been essential in the design of our study and helping us to get the word out about our research to other patients. The Count Me In team at the Broad Institute, which is really going to help us accelerate the pace of our work. And finally, our funding sources, including the Colorectal Cancer Alliance, who play such an important role in helping us to do this work. Thank you so much for your time.